I'm Maria Wilson.
And I'm Danielle Mandikian.
And we are scientists. We love science.
Yeah, we do. So when we aren't doing it, the next best thing is to talk about science. And what's really awesome is that we're surrounded by some of the most brilliant minds in research.
We're going to step away from the labs today to talk to other scientists about the cool stuff they are thinking about, working on, and imagining.
As well as how some of these discoveries just might lead to new medicines. So grab your favorite drink, get ready to unlock your science brain, and join us for Two Scientists Walk Into a Bar.
The show for scientists, science geeks, and the people who love them.
Here's the question. Where do you think the next antibiotic will come from? Do you think it'll be from a lab? Do you think it'll be found in the Amazon? Do you think it'll be found on your lunch plate?
The next antibiotic.
Yeah, probably in the ground.
Yeah, I think AI, probably mining through data and coming up with something.
Somewhere in the soil.
Lab.
Maybe another one from fungus.
My guess would be the lab. Maybe some insects.
There's going to be some AI involved with that.
Probably be a combination.
I think it's a combination of AI and lab. To her point, AI might generate the idea, but without lab intervention or lab work, it's not going to go anywhere.
Hi everyone, welcome to the show. Today we're going to take a deeper dive into the world of bacteria and antibiotics. And this is actually our 4th episode on the topic. We're bringing this back, especially this season, because the theme is unmet need. And this is a powerful example of where critical gaps in healthcare still remain. But I think we can all agree that we don't actually talk about antibiotics and the problems that come up with antibiotic resistance enough. And to kind of put that in context, back in 2023, the World Health Organization estimated that there were about 5 million deaths due to antibiotic-resistant microbes. So if we project that out to 2050, we could be on par with cancer-related deaths, and that's a serious problem we have to face. But the good news is that this is also an area where AI has many opportunities to help us move forward. So today on the show, we have Steven Rutherford, an expert in infectious disease, and Tommaso Biancalani, who's an expert in AI for research biology. Welcome to the show. So what's the deal with antibiotic discovery right now? Why is it so difficult to find new antibiotics these days?
So antibiotic discovery has been going on for many, many decades. Pharma research actually started with antibiotics. And so there are very tried, true, historical ways to discover new antibiotics. Unfortunately, most of those dried up decades ago. And so we've been trying for many, many years now to try to find new ways to kill these bacteria. One of the things that we can do is we have a really good assay to understand whether or not a molecule is going to kill a bacteria. We can ad nauseum screen new chemicals. As long as we get them in the door and we can screen them, we can see if they have activity. And so we have a pile of data where we have tested a bunch of molecules and seen this one has activity, this one doesn't, and then move on to the next one. And typically what we'll do is we'll take the ones that have activity and see if we can make them into things that can become drugs. And many times you can't, but that starting pool is really at this point our limitation. We don't have enough things that have that initial activity to be able to continue working with them to develop them into drugs.
We try to understand why is this such a difficult problem. And so what we thought is like, okay, we need to look at different molecules. The way we say it is a different part of the chemical universe, which are not in the typical small molecule libraries. But the problem here is like, how are we going to do that? How do we find a reliable way to select our drugs outside the part of the chemical space that we know? And this is where we realize that AI can really help.
Help me understand what this chemical space that we don't know is. Like, how many molecules are we actually looking at?
So the number of all possible small molecule compounds is very hard to estimate, but it's incredibly large. We think it's bigger than 10 to the 23, which is the Avogadro number, which is what a physicist would call it a very large number.
Give us a perspective. So if there's something above 10 to the 23, what we actually have on hand that we've screened is
10 to the 6th.
I'm making a grimace face.
The number of those compounds that can be active and can be used as drugs is drastically small, which is why we need AI as a compass to navigate that universe and make sure we select the right compounds in the right way.
Hi, Danielle.
Hey, Wellington.
Hey, Danielle.
What's up, Karen?
So this isn't really a question, but wow, there are a lot of needles in what sounds like a ginormous haystack.
Yeah, that's a huge number. It totally blows my mind.
Danielle, I have an actual question for you. What exactly is a chemical space?
I think the best way to think about it is what it's not. So typically we think of chemical libraries. Those are chemicals that have been identified. Someone put their hands on them at some point in time. We know they exist. Versus a chemical space is what could exist in theory. And word on the street is that there are more chemicals in a chemical space than there are stars in the universe. So I know we've covered this in previous seasons, but it's always good if you could take us back to the basics of what are bacteria?
So bacteria are ancient forms of life. They've been around forever, and they can adapt readily to every environmental niche that exists on the globe. So a couple things. One, they have haploid genomes, so they only have one copy of their DNA. And so any changes in any gene, that is their only copy of that gene. And so they can mutate and adapt through evolution to any different insults. And so any population of bacteria, there is going to be at least one bacteria among the millions that are there that's going to encode something that can provide a benefit under certain situations, including the presence of antibiotics. And so you can rapidly select for resistance. We can do this in the lab. This happens in the clinic. Every new antibiotic that gets introduced, antibiotic resistance emerges almost immediately.
A simple rule of thumb is that the smaller the organism, the faster its evolution. And so bacteria, or in general pathogen, they have very short time scales to be able to learn how to combat new drugs. And so it becomes a sort of race, like are we faster in discovering drugs than bacteria are to learn how to fight them off. And this is a very difficult problem, which is not just a scientific problem, but it's a policy problem too. For instance, the way we administer these drugs and when we use one or the other, on which context also affect the outcome.
We're really kind of fighting the clock on multiple ends, right? So I have two questions. First question is, Steve, from your perspective, would you say that AI-driven approaches are helping scientists like you learn more about the basic biology of bacteria that you might not have even looked at in the past?
Oh, absolutely, because our assay, our readout for how well a molecule is behaving is very complex. It takes into account a lot of different factors. So how does the molecule enter the cell? How does it engage its target? How does it inhibit its target? And how does that lead to cell death? All those things, we have those in one assay. But the problem is we have so many limited number of molecules that actually kill bacteria that you don't learn about some of the fundamental rules. For example, penetrance. How does a molecule get into the bacterial cell? And so if we don't have enough examples to learn what the generalized rules are, it's going to make it really hard to tease that apart. But as we expand the number of molecules we have that allow us to look at what types of things can get into a cell, maybe we can start to tease apart these rules. And that will have effect not just on the molecules we're looking at, but every molecule that anyone's ever looked at.
That's really cool. So what you're saying is that AI helps you look at more molecules than what would be feasible in a lab. And it, essentially helps you approach these questions to rules much more robustly. So this is really different from what I'm actually familiar with. Mm-hmm. So my second question is, when you're searching these questions, how are you actually approaching it? Like, what information goes in and what kind of information does it actually help you predict?
So there are essentially two different arms. So one is to leverage information from the data that we currently have or we can generate in our high throughput screen. So in that case, what's going to happen is we take bugs, we take pathogen, we expose them to compound, and then we see which compound kills and which compound does not kill. And at that point, the model learns that if you have that compound, that chemical structure, that kills and the other does not kill. So that's one important arm that the AI can learn. And we call it like a supervised model. That's very similar to the first version, or even some of the new version, even though they're more complex, of AlphaFold. That's a model that predicts the outcome of a specialized task. A second thing that we can do is to tap into our knowledge. For instance, if we have one chemical structure, one compound that kills bugs, is that compound toxic? Well, that turns out to be a very difficult question because toxicity is, we need to figure out, it's a problem that requires the understanding of the human body. And the human body is very complicated. So one way to have good answers is to tap into a large language model, because large language models are trained on human knowledge. And so they might be able to inform which compound is more or less likely to be toxic, whereas our internal data will inform on which compounds is more likely or not to kill bugs.
So you talked about kind of utilizing these large language models. Is this different from previous attempts where people are looking at like chemical libraries or genomic libraries, or it's still in that same wheelhouse of saying this was efficacious and kind of building your model that way?
So essentially, if we want to maximize outcomes, we need to take advantage of all data that the pharma company can generate, like the wet lab can generate, but then at the same time of data of a like multimodal fashion or that are on the internet. So the way to approach the AI is to use two different arms. One arm that leverages the data that are generated in-house, and that's what we call a supervised model. And then a second one is more on the foundation model or large language model side, where we leverage the public knowledge that we have. And what the model does, tries to understand which compound is a good compound, both by looking at what our data say and what is currently known.
Danielle, can you dig a little deeper into the difference between supervised versus foundational models?
Sure, as long as you can slap a disclaimer where I don't think this is my area of expertise and you fact check me. But the way I think about it, any type of model is you're putting some kind of information or data set in and you're trying to get it to recognize and put output out, okay? What's different is how big are the data sets and how are you defining those things that it's looking for. So most of the time for like kind of smaller-ish types models, you can say, here's some data, and I'm looking for X, Y, and Z to come out of it. And that's where you have supervised versus unsupervised models, where supervised is you give it a very clear example of what X, Y, and Z are. Now, when you think about a foundational type model, you're talking about a really massive data set that it's going to evaluate. And instead of you pretending like you know what X, Y, and Z are, you're going to let it figure out what X, Y, and Z are.
How does this relate to new antibiotics?
I think a really good way to think about it is if we want to come up with an antibiotic, assuming that we don't actually know what some of those rules are that are going to make it a really great antibiotic, having those much larger and complex foundational models are going to be better at providing novel answers than something where we're already saying, hey, I know what we need to look for. So this is kind of thinking about things from the discovery side, and I think you also kind of touched on predicting toxicity, so that also kind of helps to think about AI as also, kind of weighting the likelihood of success. And so how do you guys envision this closing that discovery gap or at least shortening the timeline? Like how are we seeing this play out today, or do you think that this is still something in the field that we're trying to get there and in the next 5 years or so we'll be able to see the fruit of the labor, whatever the
I think it is going to take time still, right? So the whole drug discovery process and development process still takes time, and we're still at step one of trying to find that next lead. So find that new molecule that it kills bacteria. And so once we get there, we can start the real drug discovery, drug development process and making that into a drug. But the more things we can check off from that first hit, the more efficient the rest of the steps are going to be. And so if we can build models or build in ways to mitigate as many risks as potentially possible in the beginning, that just sets us up for success later.
Yeah, so the thing about this that is such a trip is that, in order for all of this to work, data has to be usable. And there's such a big difference on how we have curated and stored data before and after AI rolled onto the scene.
Yeah, absolutely. Because traditionally, you've had biologists running these assays, and then you make a PowerPoint slide or you put it in some janky Excel spreadsheet or something like that, and then you go talk to someone who really knows what they're doing computationally, and it takes them forever to change that around and get into some sort of usable fashion. And so part of the thing that's actually enabled us as well is to be a little bit better about data curation, because we don't know how our experiments might eventually be able to feed into some of these computational models. And so if we're a little bit better about that on the front end, it'll help things in the future as well.
Tommaso?
So the drug discovery process is a very, very long one. And AI can help in two different ways. So one is to give us more hits, which means more compounds that we can consider. That does not necessarily mean to cutting the timeline short. It simply means that we have more drugs. So if it takes a certain number of years to start from a new compound to when the compound is being used, maybe with the same timeline we can just have more drugs. So the number of hits, so the number of drugs that we can find is enhance, so we find more drugs. The second thing that we can do is to try to make sure that it will take less time for us to understand whether a drug fails.
Can you expand on that just a little bit?
So in that case, for instance, toxicity is a very, it's a great case. If we were able to understand when a molecule is toxic before testing it in a human, that would be a complete game changer. Can we do that at present? I don't think so. There are many computational methods that use AI or do not use AI that can currently help. However, I do not think that predicting the toxicity of a compound is a solved problem. And so the question that we're asking right now is, why isn't it solved? And well, one basic answer is because it's a very difficult problem. But a different answer is also because we haven't been able at present to generate enough data at scale that allows those models to pick up a better signal. So better signal does not mean that they're going to be a magic wand and they're going to tell us whether a drug is toxic or not with 100%. But by generating more data, and generating data in that space is particularly difficult because it requires data on human, by generating more data, it is possible that this model will give us a better job than they currently do. And that can affect the timeline of a drug discovery pipeline. I hope I didn't say anything too much controversial here, because that can piss off.
What did you say? We need more data is what you said.
Yeah, but
we all agree. Oh, but you know, though, I mean, I think there's a true point, and again, I think it also kind of supports this idea of how critical it is to have people from different scientific backgrounds all at the table, because this idea of predicting toxicity is a huge issue in drug development, and there's a lot of people trying to work to figure out structure-activity relationships. You know, whenever we have chemical compounds, what are like, you know, consistent features that like lead to kidney toxicity and whatever, you know.
I didn't mean to imply that we need to generate data in human as in feeding compound to human and figure out they're toxic or not. I don't think that can be an implication to be taken.
It's an important point because there are some experiments we can't do. And so we can't generate a gigantic data set where we take a bunch of random molecules, put them into people and say which ones make them sick and which ones don't. And so we have to find new and creative ways to assess those in a way that's predictive. And we're not going to know that tomorrow, but if we can build those tools now, in the future, it should make drug discovery and development a lot easier.
Danielle, how successful have we been at predicting toxicity in early research, and is AI going to help this?
I think it totally depends on what molecule space you're looking at. In some cases, if you have like canonical pathways such that have already been really well defined, like in the small molecule space, I think you're much faster to success versus in the large molecule space where you have biologics, where it could be a lot more complicated and not as well defined. So where do you see some new areas of development in your field?
Yeah, and maybe I'll go back. We've been talking a lot about how bacteria are bad and they infect people and there's diseases associated with them. But I also just advocate that there are many good things bacteria do in the world that allow us to survive. And I think just understanding the basic biology of bacteria can inform many other aspects of drug discovery and not just for antibiotics. And so if we can start to tap into and really understand bacteria, how they interact with each other, how they interact with the environment, and how they interact with us humans and other hosts, we can learn a lot about biology, but we can also learn about new molecules, we can also learn about new processes, and how we can maybe even manipulate those processes by directly manipulating the bacteria.
Can you give us some examples?
The DNA of a bacteria is what tells what the bacteria should do. And so if we are able to write a DNA in a way that the bacteria would just do what we want, well, then would be particularly cool because we can have bacteria fighting other bacteria, but we can also have bacteria generating molecules that can help our immune system behave in certain ways. So to do that, we try to resort to generative AI, and we leverage the fact that we have a lot of data that tells us what a particular piece of DNA does, and we can use that to train a big model that then tries to generate a piece of DNA that makes a bacteria or a virus exhibit a certain behavior of interest.
What are some examples of this in human health where this would be really beneficial?
So one example is in cell therapy or gene therapy. So we want to design viruses that infect cells, like the good viruses infect cells and make cells express certain molecule. And one problem that we have is that when we engineer the DNA of these viruses, that is often too long. So we cannot just have a DNA which is too long because the cell would not function that way. So what we need is we need to miniaturize a part of the DNA of those viruses without losing function. So the way generative AI is helping us here is to start from a DNA which does what we want but is too long, and to take pieces out in a way that is shorter, still perform its original function.
So more to brass tacks DNA.
Yeah, we call it promoter miniaturization. And that is something we use when we need to have the expression of transgenic cargo, which are very, very, very big proteins. And because these proteins are very big, we need to make the promoter short. And if we need to make the promoter short, in doing that, the promoter stops functioning. And so the question is, how do we make the promoter short in a way that it can accommodate for the expression of a transgenic cargo? And the solution here is generative AI, and that works pretty well.
Could you give some concrete examples of how we could utilize bacteria in a beneficial way for human health?
Yeah. So I think there's biology discovery aspects and there's practical aspects. So from biology discovery aspect, we know that the bacteria that are in and on us change in disease versus healthy states. How they change is very complex and very hard to understand. But if we could understand that, it might give us insights into what is causative for disease or what is propagating disease. And that's not infections only. That's any inflammatory disease, for example, or any immune-related disease. And so if we can start to understand that, we can gain some new insights and potentially develop targets. The other way is completely leaning on the historical precedents for manipulating bacteria themselves. And so if we have a specific process that we want to carry out, but there's no other way to access that process other than having a bacterium and get to a specific spot in your tissue, we can actually engineer those bacteria to do functions that we think are critical. So very much like these viruses that Tommaso was talking about, we can have the bacteria go and serve as factories for delivering drugs, for example. And that's not science fiction. People have done that historically. And if we can find the right targets, the right molecules to deliver, and the right bacterium to do that, you can actually make a drug out of them, potentially.
Yeah, that's really exciting because there's a lot of like gene therapies where there's always that fear that you're going to have like a misinsertion that causes another disease. So having something that you could eventually kill out would be really great for gene therapy.
And think too, like with these, you're not necessarily limited by just delivering one molecule and having the patient take that. You could have bacteria encode multiple processes or multiple drugs within them.
One question. When I say bacteria fighting other bacteria, is that accurate or is that not?
I mean, in nature, we know that bacteria combat each other, and that's how antibiotics likely came about. It wasn't because they were developing drugs for humans, it was because they want to compete with the partner.
So if I said use generative AI to have bacteria fighting other bacteria, which is not something we are doing, but
no, it's not.
It's not science.
But again,
it's not a controversial statement.
We should talk about this more. Is like because antibiotics as natural products, which is very different from the types of molecules we've been talking about. Those have served humanity very well because we've been able to use those to treat patients that otherwise would probably die by bacterial infections. If we could figure out what the code is to figure out all these natural products and dig for new ones, that'd be great, because this is exactly what Tommaso was saying earlier. We've just been retesting the same thing over and over again in soil, and you get the exact same antibiotics over and over again. So we've already picked out the low-hanging fruit. We need to climb higher to be able to get those more high-hanging fruit that have been just harder to access. And people have tried this. There's been evidence in the scientific literature about people have found new ways to probe these natural products, but we have to find some other better ways. And there's many untapped sources for these that if we could crack that code, that would also open up new drugs.
I'm super curious about, like, one of the things that I think is the most fascinating about all of that too is also like the environmental selective pressures that trigger them to then release whatever chemical warfare they want to unleash between each other. And I think it's also interesting, something about like the environmental aspect of it is also very interesting too, for even thinking about what we would want to target. Because sometimes the thing that's crazy is like you have to have these you generate these antibiotics and they're not specific against a single bacteria. They hit up groups. It's like whatever promiscuity you can bank off of. But it also can wreak havoc on all of your beneficial bacteria. And so anything to kind of like tighten that up would be really interesting.
Yeah, and that's something that's I guess evolving in the field as well. As people are trying to find new molecules, one of the things that we test now that we wouldn't have even 5 or 6 years ago is, hey, how does this molecule behave against the typical gut microbiome, and what is the impact on that? Because we know now antibiotics, even early in life, have long-lasting, almost permanent effects on the microbiome of patients. And because we don't know how all that affects all these different intricacies of human health, we want to make sure that we're not doing something early on that's going to have a dramatic effect later in life.
Danielle, definition, what is a promoter?
Good question. So whenever we're thinking about, and especially in the context that it was discussed here, we have information that's stored in our DNA, and we have to take that information and somehow get that into protein form, right? And so one of the steps that is needed for that to occur is that we have a sequence of DNA. A promoter is a very specific part of that sequence that is necessary for all of the proteins and pieces to come together that are going to then be able to transcribe part of our DNA code into a mRNA transcript that is then going to be, go to another part of the cell, be translated into protein. This episode's a lot of fun because we're getting to bring up all of these basic players for cell biology. And so when we're talking about promoters, what we're really kind of talking about is kind of the players that are first involved when we have a DNA sequence that is later going to be a section of it copied out into an mRNA transcript, and then it moves over and is translated into protein. But in order for all of that to kick off, what's really critical is that you have a promoter that says, hey, start at the DNA transcript here, and this is where you're going to start, making that functional copy. And so whenever we were talking about it in the sense that we were on the show or this episode, what we're really talking about is sometimes you depending on like how big of a protein that you need to come off of this, you may have to play around with the total size of the promoter, and there's ways to do that. What about the business side of this actually makes it challenging on top of the science?
Antibiotics are critical to human health. They're critical to prevent life-threatening infections, but they're also essential for things like immunosuppressive therapies or surgeries where there's potential for infection. And so we need to have an armament of antibiotics to combat those infections. The situation right now is it's really hard to, let's say, economically justify going after antibiotics, though, because they are not these blockbuster drugs.
Yeah, no, just to reiterate what Steve just said, antibiotic discovery is to some extent the anti-business, because if we find a new drug, the last thing that we want is to use it massively. Because the reason is that when a patient is sick, you first want to give drugs that we already know, because every time we administer these drugs, the bacteria or like pathogens in general develop a resistance to that drugs. So if we discover a new antibiotic, we want to use it as a last resort only. But that means that we use it less, and so we sell it less. And so this disincentivizes businesses into going into antibiotic research, despite the fact it is such an unmet need.
And that shouldn't also downplay that it's also hard. There's not easy science to get to discovering antibiotics. But there are pushes to have incentivizations both for doing the research as well as the development and marketing those drugs. But yeah, it's going to require the entire country and world actually to get behind some of these initiatives.
Yeah. What's your estimate for 2050? What's up with AI?
Or you've solved all the antibiotic crisis.
You've solved the antibiotic crisis.
What are you doing next?
I wish I knew it. There's a lot of dystopian TV shows that give us a nice perspective of what can happen.
Those are the ones that didn't discover antibiotics, though. Right?
No, those are not. So one of the coolest things that AI can do these days is to use tools. So for instance, when you write an email, you open Gmail or some other tool and you just type it. And you can also, play a video game or use software or use words. And all these activities done by human can also be done by other software. And this is what we call agents. Agents is software that can access other software. And the important part is that they can reason. I use this word. It's actually fairly different than what that means for the human brain, but the net effect is sort of similar. So they can reason of what the data and software say. So what people are looking into now is to build agents that can access our data and the internet too, and other tools. And then they try to build an automated reasoning platform that can understand, by combining all these different options, what is the most sensible scientific way to move forward.
I didn't know that that was the word for it, and we have been fighting with this ourselves in our own group. It's just like, it's kind of funny, like everything I feel like is coming so fast and everyone's trying to like catch up and figure out how to utilize it properly.
The word agent?
Well, no, just like everything with AI. It's like there's so many opportunities, it just feels like it's like an explosion of stuff.
I think it's an explosion, and then it's probably gonna plateau at some point. But now we are still in the explosion phase. So something similar that happened was when we were buying computers. There was a time in the 80s and the 90s too, when we were buying computers, that every year there was a new kind of computer that was faster than the other one. Whereas now, we still have computers that are better than the one in the previous years, but the changes are not as big. And well, it's very hard to say whether this is going to happen for AI as well, but I do think that at some time, there will be a plateau of what AI can do.
So just to make sure I'm tracking you, I'll definitely ask you as well, but so are you saying that you think in 2050 we'll be in that plateau and all of these agents will be able to run and integrate things very efficiently?
It's very hard to be concrete I'm
not going to hold you to it. I won't come knocking at 2050 and be like, hey.
Yeah, I don't know.
Fair enough.
Yeah.
Cool. What about you? You got your 2050 prediction?
What our understanding of what AI brings, having worked with Tommaso and his group, is that the things that might have taken us a long time are taking us less time. And that's great for the idea of making new antibiotics, because we need more. We're going to continue to need more. We're going to continue to need to discover more. But what it might do then is if we can be more efficient and quicker about that, it might open up our time to start thinking about these other things we've talked about. What other uses of bacteria are there? What other things can we do? What other types of bacteria can we build? And if we don't have to worry about dying from infection, it opens up the time to do those types of things. That's the hope, right? The hope is that those dire predictions for 2050 don't come true, because we or somebody else finds a way to combat this.
Thank you so much. This has been really interesting talk. It was really great to meet both of y'all.
Oh, thanks
so much. Yeah.
Danielle, great conversation. Even though the task seems really daunting, I came away thinking that AI is really going to help us sort through what seems like a galaxy of possibilities. How do you think this is going to cross over into your field?
That's kind of a funny thing, because sometimes these types of crossovers are so unexpected. I know you like a backstory, so I just, I wish I could claim fame to this, but I can't. But one of my favorite stories I ever heard was about this scientist who really loved scuba diving. And one time he was underwater taking pictures of a coral reef, and he found this glowing eel, which was really special because we didn't know invertebrates made proteins that glowed, right? These fluorescent proteins. He spent a whole career, short version of the story, he eventually found that eel, took it back to his lab, froze it, chopped it up, identified that fluorescent protein, and it completely revolutionized the types of readouts that we were able to get on ion channels. So he did that vertebrate. What made it really special is that fluorescent protein was smaller than one than we'd ever known before. And because of its size, he could stick it on ion channels and we could visually watch networks of neurons signaling with depolarization. And so when I think about how AI could really kind of cross that space, like how lucky were we that there was a dude who loved scuba diving and happened to also like photography and happened to be a badass biochemist, right? So the same kind of way that I think that this could cross over is, what are the types of molecules that we're going to find in this chemical space that we would have never gotten our hands on otherwise? How could we utilize those, not necessarily just for antibiotic resistance, but maybe those will become parts of smaller libraries and those potential hits that we might utilize those molecules for something else in life sciences research that we would have never thought of before. It's really, what's this, serendipitous thing that we're able to find?
So Danielle, are you talking about molecular design here?
Absolutely. Using AI to give you access to this chemical space, it's really letting us see molecules we've never seen before, and we can think about utilizing the function from those molecules in novel ways. So you really are getting at that molecular design by new molecules, new function, new applications. I think what's going to be really exciting is seeing how does this impact antibiotics discovery, antibiotics resistance, but also all the other different places in life sciences that can benefit from it. I mean, this is cool. And that's our show. Thanks so much for listening. If you haven't already, rate our podcast wherever you listen. It'll help new people find us. And make sure to subscribe. If you have any questions about the show, you can contact us at podcast@gene.com. And now for me, it's back to stocking cells.